A very good evening to all of you. Just a moment. Right, I hope you can see the screen, right? Can you see the screen and hear me? I just want to know that whether I am audible and the screen is visible. Can anyone put in the chat box?
Whether I am audible and the screen is visible. Can anyone put in the chat box? Whether I am audible and the screen is visible. Can anyone put in the
Oh, thank you, Prathamesh. So, without wasting much time, as we saw in the morning, right, we'll be talking about some MCQs related to your MSc microbiology entrance examination. So, you're here with me, Professor Girish Kukreja. Yeah, so the first one, I know you all are tired. Some of them are eating food. Some of them must have had food, and I'm also still going to have my food. Anyways, that is not the question. The nucleic acid sample. Now you're having a nucleic acid sample, that is, say, a DNA sample, and this is not having a ratio of T plus C is equal to A plus G. Oh my goodness. A DNA sample must do this, right? A DNA sample must have A plus G is equal to T plus C, right? The number of purines is equal to the number of pyrimidines, what we say. So now this is a sample and this doesn't have this A plus T. Oh, not A plus T. It is A plus G, that is purines is equal to T plus C. You know, that is what is referred to as the Chargaff's rule. So the DNA, it is double stranded and the two strands, they are complementary. A pairs with T, G pairs with C, right? So you have A pairing with your T and G pairing with your C. Therefore, the number of your A plus G is equal to the number of T plus C, the Chargaff's rule. This is not happening here. This may be because it is AT rich. No, it will remain the same. It is GC rich. No, it will remain the same. It is double stranded. Rather, in double stranded always occurs. Where it doesn't occur is a single stranded DNA. So the DNA is single stranded, right? Maybe then this, not maybe, then for sure A plus G is not equal to T plus C. So if a nucleic acid is not showing the Chargaff's rule, maybe it is a single stranded DNA. Anyways, a fatty acid of 16 carbon having dash has a high melting point, right? So fatty acids, if you remember, they are classified into two. One are saturated and other as unsaturated fatty acids, right? So your fatty acids, basically you know that they are long chain hydrocarbons. So you have CH3, CH2, 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 a lot of CH2s of them. And then at the end, you have what is called as COOH, right? So fatty acid of 16 carbon atoms, right? Can you tell me which is a fatty acid, which is a saturated fatty acid, a common fatty acid, which contains 16 carbon atoms? Anyone of you can knows the name of a 16 carbon fatty acid? Yes, anyone there knows the name of a 16 carbon fatty acid? If you thought I have gone somewhere, I am still here. I was looking at this phone like if I'm getting any answers right there. So a 16 carbon fatty acid. Anyone knows a 16 carbon fatty acid with a saturated kind of a thing which has only single bonds? A very common 16 carbon fatty acid. You don't know the name of a 16 carbon fatty acid? Can anyone type it? Wonderful. Yes, it is palmitic acid, Priyanka. It is not palmitic. Anyways, the typing mode is alasel, I know. It is palmitic acid. Great. So, yes, great, Prathamesh. It is not glyceride, Prathamesh. It is palmitic acid. Okay. A fatty acid of 16 carbon has dash high melting point, right? Which fatty acid will have a high melting point? The one which has a double bond, the one which has only single bonds, the one which like has multiple double bonds, the one which has two double bonds, or it does not depend on bonds, right? So here we know that fatty acids, if they are saturated, that is if they are having only single bonds, right? So they all will be like properly oriented and packed very close to each other, right? So when they are all saturated, having only single bonds, the molecules are like very close to each other. They are packed closely to each other, right? And if you have an unsaturated fatty acid, probably wherever the double bond is present, there is a bend or a kink in the molecule, right? So जितने ज्यादा आपके fatty acid में double bond होंगे, उतने आपके molecule में bends होंगे. So because of those bends and kinks, the molecules are away from each other, right? So if it is having only single bonds, if it is only a saturated fatty acids, the molecule are very close to each other. And when the molecules are very close to each other, obviously you will have to supply a lot of energy to separate them, right? Going from a solid state to a liquid state, you will have to supply a lot of energy, and therefore it will have a high melting point. So the one which is having all single bonds, that will have a high melting point. Can you tell me A, B, C, D now? Who is Raj here? I'm not Raj, I'm Girish. Yes. Which one now? याच्यातलं सांगा आता की two double bonds A, B, C or D. The one which has high melting point. Which one has a high melting point? A, B, C or D? Which one? I hope you can read it. A is two double bonds, B is only single bonds, C is does not depend on bonds, and D is multiple double bonds. At least comment मध्ये A, B, C, D काहीतरी टाका. Yes, please, which has a high melting point. The more single bonds are there, the double bond will introduce a bend in the molecule, so it should have only single bonds. So it is A, B, C or D out of these. Priyanka says D, multiple double bonds. Nahi, जेवढे double bond just असतील, right? The more are the double bonds, you will find that the molecule will be more separated from each other and the melting point will be low. It should have no double bonds. It should have only single bonds. Now tell me the answer. Yes, now Priyanka says A, no. Two double bonds, nahi. Please tell the right answer. Yes, anyone else there? Anyone else there? Fatty acid, which one will have a, I'll again tell you, if they are very close to each other, if they are having only single bonds, only then they have a high melting point. याच्यातला high melting point कोणता असेल? I'm still waiting for your answer. Till you give me, I'm not going to go. येस, ज्यांच्यामध्ये फक्त सिंगल बॉन्ड आहे, दोज विच हॅव ओनली सिंगल बॉन्ड्स दे विल हॅव अ हाय मेल्टिंग पॉइंट. सो इट इज A, B, C or D. मी जाणार नाही. टेल मी, यू नो आय एम व्हेरी stubborn in that case. Yes, Prathamesh, Vivek, Priyanka, Patil, anyone can tell a fatty acid which one will have? Yes, Priyanka, thank you so much. पुढे जात आलं नसतं. It is your B which has only single bonds. Thank you so much. Common component or building block of phosphatidic acid. A common component or building block of phosphatidic acid and phosphatidylcholine is, right? Which of these? Thank you so much. Thank you, Raju. It is not D. Press Pratik. It is B. Yes, Vivek. It is B. Right. So multiple double bonds will not have a higher melting point. They will have a lower melting point. Let me come to the third one. The common component of phosphatidic acid. आठवतंय का फॉस्फॅटिडिक ऍसिड? फॉस्फॅटिडिक ऍसिड मध्ये काय असतं? लाईक टिपिकल फॉस्फॅटिडिक ऍसिड विल कंटेन अ ग्लिसरॉल बॅकबोन. इट विल कंटेन 2 fatty acids and a phosphate group, right? Let me take a break for a moment. So, but just let me tell you, this has a glycerol molecule. We will take a short break of 1 minute, right? We are in the short break. Please stay tuned with me. Till then, you can read the questions. At least you will come to know what is going on. It's okay. It's okay. Let's have some patience, Prasanna. Right? Just a moment. It's almost done. Yeah. So phosphatidic acid, right? You know a phosphatidic acid. वापस एक बार आ रहा हूं मैं. Phosphatidic acid में एक क्या है? ध्यान से सुनो यार, प्लीज. Phosphatidic acid में एक क्या है? एक ग्लिसरॉल है, 2 फैटी एसिड है और एक फॉस्फेट है. एक ग्लिसरॉल, 2 फैटी एसिड और फॉस्फेट. और फॉस्फेटिडिलकोलिन में क्या है? एक ग्लिसरॉल है, 2 फैटी एसिड है, एक फॉस्फेट है और एक हेड ग्रुप है, राइट? तो व्हाट इज कॉमन? आप अभी बता सकते, मैंने अभी जस्ट बताया. इन दोनों में क्या कॉमन है? ग्लिसरॉल, लाइसिन, कोलेस्ट्रॉल या फ्रुक्टोज, इन दोनों में क्या कॉमन है? व्हाट इज कॉमन इन फॉस्फेटिडिक एसिड एंड फॉस्फेटिडिलकोलिन? आई जस्ट टोल्ड. You have to be present there. Yes, पाटिल, प्रतीक, विवेक, प्रियंका, प्रथमेश, दोन्ही मी आता जस्ट सांगितले. फॉस्फेटिडिक ऍसिड मध्ये परत एकदा रिपीट, आय एम रिपीटिंग वन्स अगेन. इट हॅज ग्लिसरॉल, 2 फॅटी ऍसिड and a phosphate, and phosphatidylcholine has a glycerol, 2 fatty acid, a phosphate and a head group. So what is common? Yes, it is glycerol. Great. So you have glycerol. Glycerol, you all know the structure of the glycerol. It is trihydric alcohol, right? So glycerol looks like something like this: CH2OH, then you have your CHOH, and then you have your CH2OH, right? fatty acids are esterified to it, right? So there is an ester bond between them. Great. So here you have what is called as your glycerol. Let me go ahead. The vitamin component of coenzyme A, right? You know, coenzyme A, your vitamins, they are converted into coenzyme forms. Like you have your vitamin B6, that is your pyridoxine, that is converted to your pyridoxyl phosphate or pyridoxamine phosphate, right? You have niacin, which is converted into NAD, nicotinamide adenine dinucleotide, right? Then you have your riboflavin, right, which is converted to your FMN, or it is converted to your FAD. So vitamins are converted to coenzyme forms. So here, so you directly have coenzyme A. Can anyone tell like what is the vitamin component of coenzyme A? कौन सा विटामिन है? इसका ऐसा है कि वो बीच में है वो विटामिन का कंपोनेंट. कैन एनीवन से विटामिन कंपोनेंट ऑफ कोएंजाइम में कौन है? एस्पार्टेट, इट इज पीएलपी, इट इज बायोटीन, और इट इज पैंटोथेनिक एसिड. Remember the coenzyme, remember the vitamins and the coenzyme forms. I told few of you, right? मैंने अभी थोड़े दिन बताया, जैसे नाइसिन का NAD है, राइबोफ्लेविन का FAD है, राइट? फिर आपके हम बोलेंगे कि ये कौन था आपका बायोटीन का बायोसाइटिन है. Anyone knows about this coenzyme A, which acts as a carrier of acyl groups, you know, that acetyl CoA है ना? तो वो CoA जो आपका आता है, that is coenzyme A. उसमें कौन सा विटामिन है? Anyone knows that? No, Raj, it is not aspartate. For your kind information, aspartate is not a vitamin. You know that aspartate is an amino acid. Can anyone tell what this aspartate is? It is a acidic amino acid, a basic amino acid, or a neutral amino acid. Now I am becoming like the one which takes that viva voce. तो आपके जो examiner viva लेते वैसा हो गया थोड़ा. But anyways, aspartate आप बता सकते हैं? Yes, प्रियंका, it is pantothenate. The answer is right. कोएंजाइम ए का जो विटामिन फॉर्म है वो कौन सा है आपका? पैंटोथेनेट. बट नाउ आई वांट टू नो, सिंस राज हैज टोल्ड, can you tell me that aspartate is which amino acid? Like it is an acidic, basic, neutral, aromatic. Which type of amino acid is aspartate? Aspartate is which type of amino acid? Acidic, basic, neutral. कौन सी type की amino acid है aspartate? Anyone of you out there? Aspartate is which type of amino acid? It is acidic, it is basic, it is neutral, or it is aromatic. Aspartate. नाम ही यार, एस्पार्टेट को हम क्या बोलते हैं? एस्पार्टिक एसिड. नो, इट इज नॉट एरोमेटिक. लक्षात घ्या, एरोमेटिक अमाइनो एसिड. एरोमेटिक अमाइनो एसिड कोणती आहे? फिनिल अलानिन टायरोसिन ट्रिप्टोफॅन. दीज आर एरोमॅटिक अमाइनो एसिड्स. फिनिल अलानिन टायरोसिन ट्रिप्टोफॅन. परत एकदा सांगतोय, कोणाला सांगू नका, डोंट टेल एनीवन. फिनिल अलानिन टायरोसिन अँड ट्रिप्टोफॅन, दीज आर अमाइनो एसिड. आई वांट टू नो एस्पार्टेट. Yes, anyone out there? Aspartate. Prasanna, you're still there? Or Prathamesh, Raj, anyone wants to tell this aspartate is which type of amino acid? Acidic, basic, neutral. It is not aromatic, but aspartate को दूसरा नाम क्या है? एस्पार्टिक एसिड. अभी तो आप बता सकते हैं कि वो कौन से टाइप की है? Acidic. Thank you so much, Raj. Thank you so much. It is acidic. Identify the odd one out. I'm not taking much of your time. I am too hungry. So you have this one. Yeah. So identify the odd one. You want to find out the odd one in this. The mitochondria, the nucleus, the chloroplast, and lysosome. Let me help you out with this. Like if I am drawing mitochondria, how I draw? This is the outer membrane. Then this is the inner membrane, the cristae. If I'm going to draw the nucleus, this is the outer membrane, this is the inner membrane. It has those nucleus, right? And if I'm going to draw chloroplast, this is the outer membrane, this is the inner membrane. And if I'm going to draw the lysosome, this is the lysosome. Now tell me the odd man out. कितना simple कर दिया मैंने आपको अभी. Which is the odd one? Which is the odd one? अभी तो मैंने कितना simple कर दिया आपके लिए. Mitochondria, न्यूक्लियस, क्लोरोप्लास्ट और लाइसोसोम. इसमें अलग कौन है? विच वन इज द ऑड वन? ABCD भी आप लिख सकते हैं. ABCD, CDEFG. एनीवेज, यस, ग्रेट. थैंक यू सो मच. Thank you so much for this. Right. So lysosomes, they are bound by only one membrane. So they are bound by only one membrane. So therefore, it is the odd one out. Thank you so much. So lysosomes, yeah, lysosomes, they are the odd ones because they are having only one membrane. You know, mitochondria is called as the powerhouse of the cell. Nucleus is called as the brain of the cell. Use some of it, right? Chloroplast is the kitchen. They want to doing photosynthesis, and lysosomes are the suicide bags, right? Having hydrolytic enzymes, right? So it is your lysosome. Yes, Prathamesh, it is D, and yes, Vivek, it is D. It is lysosome. Okay, the bivalent. Let me clear this now. The bivalent, right here, you can see. Oh, do you really? Is doubtful, is it? Because bacteria, they do not undergo mitosis and meiosis, right? But then yes, Pratik, it is D. Bivalent in prophase 1 is 10. The chromosomes in anaphase 2, oh my goodness. Bivalent कसा दिसतो आपण बघूयात. Like when the cell is preparing for division, bivalent कैसा दिखता है? देखो ये एक और ये दो. ये ऐसा जो स्ट्रक्चर आपको दिख रहा है ना, दिस इज बाइवेलेंट, राइट? सो दिस particular structure is what is called as your bivalent, right? So this is how a bivalent looks like in prophase 1 of meiosis 1. So this is 10. Oh my god, ये एक bivalent हो गया, right? ये एक है bivalent. ऐसे कितने हैं उन्होंने बोला है? 10. पहले हम एक का देखते क्या होता, फिर बाद में देखेंगे. जैसे ही meiosis 1 खत्म होगा, this will appear something like this, right? Get separated. The homologous chromosomes will get separated. When it goes in anaphase 2, these will also get separated, right? Right. So this is how it will look in anaphase 2. So to sum up, एक बाइवेलेंट के 4 क्रोमोसोम हो गए. अभी सिंपल करते हैं हम. एक बाइवेलेंट के एनाफेज 2 में कितने क्रोमोसोम्स हो गए? 4. 1 बाइवेलेंट इज 4 क्रोमोसोम्स. अब देखते हैं किसका मैथमेटिक्स अच्छा है. 10 बाइवेलेंट इज हाउ मेनी क्रोमोसोम्स? 1 बाइवेलेंट इज 10 क्रोमोसोम्स. 1 बाइवेलेंट 10 क्रोमोसोम्स. 10 बाइवेलेंट्स हाउ मेनी क्रोमोसोम्स? एवढं अवघड मॅथमॅटिक्स विचारलं का मी? 1 bivalent, 4 chromosomes. 10 bivalents, how many chromosomes? 40. Great. Yes, it is 40. So bivalent here will have how many chromosomes? 40 chromosomes. Thank you so much for those who are answering. The ultimate electron acceptor in lactic acid fermentation. I should have a big board, right? You can gift me one. I'll send you a postal address. The ultimate electron acceptor in lactic acid fermentation is dash. Now, I hope you remember the lactic acid fermentation. The lactic acid fermentation, you remember lactic acid fermentation? You start with glucose, right? Then glucose gets converted to pyruvate. And this pyruvate gets converted to lactate. तो यहां पे जो आपका NAD gets converted to your NADH, that is being accepted by pyruvate, and NADH is getting converted back to NAD, which can be reused in glycolysis. तो मतलब ये जो NADH के इलेक्ट्रॉन आ रहे हैं यहां पे, ये कौन एक्सेप्ट कर रहा है? ये pyruvate. So ultimate electron acceptor in lactic acid fermentation, where lactate is being formed, is your pyruvate. तो इलेक्ट्रॉन जो ग्लाइकोलिसिस में से बाहर निकले थे वो एक्सेप्ट किसने कर लिया? आपके पायरुवेट. और पायरुवेट का क्या हो गया? लैक्टेट. ग्लूकोज कितने कार्बन का था? 6. पायरुवेट कितने कार्बन का है? 3. कैन एनीवन टेल मी लैक्टेट कितने कार्बन का है? Can anyone tell me lactate is of how many atoms now? Glucose was of 6 carbon atoms. Pyruvate is of 3 carbon atoms. Pyruvate is reduced by lactate dehydrogenase to form lactate. So can you tell me lactate is made of how many carbons? 1, 2, 3, 4. How many carbons is lactate made of? Any one of you out there? Pratik, Prathamesh, Raj, Priyanka, those who are answering, Vivek. There are many more who are joining. Yes, Raj, wonderful, wonderful. Thank you so much, Raj. It is 3. Lactate is of 3 carbon atoms. Can anyone tell during lactic acid fermentation, carbon dioxide is released or not? Yes or no? Type Y or N? लैक्टिक एसिड फर्मेंटेशन में कार्बन डाइऑक्साइड रिलीज होता है या नहीं? यस और नो, वाई और एन? in lactic acid fermentation is yes or no? Yes, please. It is yes or no. In lactic acid fermentation, there is release of carbon dioxide. Yes or no? No. Thank you so much. There is no release of what is called as carbon dioxide. Okay, let's come to the next one. The site of photochemical reaction, that is your photophosphorylation, where you are converting the light energy into chemical energy. What is the site of that particular reaction? Obviously in chloroplast. We are talking about the eukaryotic cell now. It is in the thylakoid lumen, or it is in the chloroplast stroma, or it is in the thylakoid membrane, or the inner membrane of the chloroplast. Let me be more clear in this. So like if we are talking about a typical chloroplast, they are talking about the light reaction. So chloroplast should be green, right? Let me check if the green marker is working. So you'll have the feel of that chloroplast. So you have a chloroplast here, right? So this is the outer membrane, this is the inner membrane, and this is the thylakoid membrane. So the question is कि आपका लाइट रिएक्शन कहां हो रहा है? Thylakoid lumen, right? So this is the place A, thylakoid lumen. Chloroplast stroma, that is B. Thylakoid membrane, ये वाला membrane C, या फिर inner membrane of the chloroplast D. अब बता सकते हैं कहां पे हो रहा है लाइट रिएक्शन इन सब में से? A, B, C या D? Thylakoid lumen के अंदर, chloroplast stroma के अंदर, thylakoid membrane के अंदर, या inner membrane के अंदर? Where is the light reaction taking place? Yes, Priyanka, it is C. Yes, it is in the thylakoid membrane. तो याद रखो यहाँ पे आपका ये जो लाइट रिएक्शन है वो सारा कहाँ पे हो रहा है? In the thylakoid membrane. Thylakoid membrane is the site for the light reaction. Great. So light reaction का site क्या है आपका? Thylakoid membrane. GUG as the starting codon codes for, right? GUG, AGOG, logi, sonoji. Anyways, GUG, the GUG. So GUG, if it is the starting codon, it codes for what? It codes for valine, cysteine, methionine, or isoleucine. Comment A, B, C, or D. Oh, where is the C? So it is A, B, C, D. When GUG is the start codon, generally the start codon is AUG, right? But if at all उसकी थोड़ी कमतरता हो गई है तो अगर AUG नहीं है और GUG है स्टार्ट में प्रोटीन सिंथेसिस के, तो वो क्या कोड करेगा? वैलिन, सिस्टीन, मिथियोनिन और आइसोल्यूसिन क्या कोड करेगा? लाइक व्हाट इट विल कोड? Yes, anyone out there? GUG, if it is the start codon, it will code for what? Valine, cysteine, methionine, isoleucine. Anyone there? Priyanka says isoleucine. I'm waiting for some more answers. I'll not say whether right or wrong, but I'm waiting for some more answers. Jagtap says A, valine. Okay, Vivek says A. Anyone has a different opinion? You have so many opinions, but then opinions can be right or wrong. I'm not talking about opinions here anyways, but the facts are only one. You may have your opinion whether the sun should rise from the east or it should rise from the west, but the fact is that it rises from the east. Yes, Vivek says A, Priyanka says D. Anyone wants to say anything else? The platform is free for you all to say. Yes, GUG as a starting codon, what it codes for? GUG as the start. अरे यार start codon बोला है ना. Always it starts with which amino acid? Yes. Anyone else? It starts with any protein. It starts with which amino acid? Prathamesh says no. No म्हणजे काय आता कडे? No म्हणजे माहित नाही असं म्हणतोय का? It starts with which amino acid? Protein is always going to start with which amino acid? It's always going to start with methionine. Mala's book lagli, so I have to just make it fast. So it is going to start with methionine. Yes, Priyanka, it is going to start with what is called as your methionine. So if it is in between, if the protein is in between, probably it will code for valine. If that GUG is in between, but if that GUG is at the start, it has to code for what is called as your methionine. Okay, the last one there now. No, it's not stroma. It's okay. Ecosystem involves recycling of all except. इकोसिस्टम मध्ये सगळ्यांचं रिसायकलिंग होतं, फक्त कोणाचं रिसायकलिंग याच्यापैकी होत नाही. वॉटर, कार्बन, एनर्जी आणि नायट्रोजन. What keeps on coming back and goes back and comes back? What doesn't come back or doesn't go back? Maybe it goes in only one direction: the water, the carbon, the energy, or nitrogen. What do you think like keeps on flowing in one direction only? It doesn't go back like this. It goes only in one direction like this, right? कौन सी चीज एक ही डायरेक्शन में जा रही है? साइकिल नहीं होती इकोसिस्टम में. Which thing? Yes, it is energy. So energy goes only in one particular direction. Okay. Now it's time for some aptitude question. I'm going to take only one. खूब दम लो याता. Remaining things we'll see tomorrow. So here you have one question out there. You have to find the next one in the series. You have 7, then you have 15, then you have 31, then you have 63. Can you tell the next one? Yes, Raj, it is energy. Can you tell the next one in the series? 7, 15, 31, 63. What will be the next? Don't Google lagao, akal lagao. Someone will put it in the Google and they'll get the answer. Don't do that. 7, 15, 31, 63. Because जिंदगी में कहीं ऐसी जगह अटक गए जहां पे इंटरनेट नहीं तो दिमाग चलना चाहिए. इंटरनेट चले ना चले, this should work. Tell me this. This is energy here. The answer is energy here. This one, 7, 15, 31, 63. Can you guess the next one? This is the last one for you all. Yes, wonderful, Raj. It is 127 here. So it is into 2 plus 1, right? So 7 2s are 14 is 15. 15 2s are 30 plus 1 is 31. 63 2s are is 126 plus 1 is 127. Hush. So thank you so much. My class is done for today. We almost did 10 MCQs. We did 10 MCQs in the morning also. So we are almost done with 20 MCQs by now. I would just like to know that whether it's possible for you to join tomorrow again at 7 AM like you joined today. सुबह अभी बारिश हो गई है, वातावरण थोड़ा अच्छा हो गया, तो आप निवांत सो जाएंगे. तो सुबह उठेंगे कि नहीं आप बताओ, नहीं तो मैं उठूंगा, आप नहीं उठेंगे तो मैं पढ़ाऊंगा किसको? So just put a thumb or say yes or just put Y if you're too lazy कि आप सुबह 7 बजे ज्वाइन हो सकते हैं क्या नहीं. Because I have a class at 8, so I don't have any other option, right? And I have classes continuously, so it's possible only at 7. So for how many of you it's possible at 7? Pratik, it's not 130, it is 127. मी करून दाखवले into 2 plus 1, right? सकाळी 7 वाजता, tomorrow morning at 7. Is it possible for you all? Please tell me in the comment box. I'll be waiting for your comments, and now I am leaving. Thank you so much. Bye-bye. Good night. Take care. And someone will tell you have sweet dreams. Yes, Priyanka says it's possible. Sakshi also says it's possible. Bakichanna nahi hai what possible. They are going to sleep. Anyone else? Is it possible? Yes. Shraddha also says it's possible. So tomorrow morning at 7, So should we finalize then? So yes, I think we have 4 years, only one more years, and then I'll finalize. Only one more years we can leave. Only one more years. Vivek says yes, Priyanka, Sakshi, Shraddha says yes. आता हे सगळे येस म्हणतात ते सकाळी आले पाहिजेत. आय बी चेकिंग वेदर दीज पीपल आर रिअली कमिंग अप ॲट 7 AM in the morning. Only one more yes for me to stop this session. मलाच टाईप करून टाकावं लागेल वाटतं येस, नाही तर माझ्या मुलाला सांगावं लागेल येस टाक. Yes, Vivek, Priyanka, yes. Pratik says yes. Thank you so much. I would like to actually know, I told in the morning also that you please type your name and your place from where you are so that I come to know from where you have joined. If you can at least put the name of the state or the name of the city in the comment box, I would like come to know from where you are exactly. Right? Thank you. Bye-bye. Thank you so much. My laugh is only good. My teaching is not good. Yes, please comment the name of your city or state and then you can leave. Thank you so much. Bye-bye. Good night. May I know who this Patil FF is? Krishna, can you please get me a glass of water? Krishna is my son, don't worry. Thank you. Patil, you are from where? Prasanna? So in the morning also actually I gave my number. I'll share it once more. If I'll be sharing it like the live sessions on my status. You can just text me. It is 9823683317. Right? So you can connect me here, put me your name so that I'll save it, and for the next live session you can be here tomorrow morning at 7. It is 9823683317. Bye-bye. Okay. Okay, Prasanna. So you have to please connect me on WhatsApp. It is 9823683317. Thank you and good night to all of you. Bye bye.
प्रथमेश थांबूयात, सकाळी घेऊयात बाकीचं. प्रथमेश, विवेक,